Hello, my name is Ryoko Oi. I'm a researcher at G&L Kyosei Institute. This time I introduce protein 0th-order structure to be related to GADB protein water hypothesis by Dr. Kenji Ikehara. I'm in charge of this narration. We know a route for creation of a new protein homologous with previously existing proteins in a family. It is gene duplication theory proposed by Dr. Susumu Ono. He predicted that after duplication of a gene, one duplicate might acquire genetic information encoding a new protein of which function is necessary to adapt to new environment, while the other duplicate retains the original genetic functions. This theory is quite important as a molecular mechanism for creation of new proteins, but this route always requires a previously existing gene to create a new gene in a family. So this doesn't explain the way how to create the first ancestor gene in the family. I propose GC-NSF hypothesis on formation of entirely new gene and protein. This hypothesis explains that entirely new gene and protein was generated from non-stop frame on antisense strand of GC-rich gene after gene duplication. Only the GC-NSF out of 3 codon sequences on antisense strand is a base sequence quite different from the GC-rich gene on sense strand, but has similar base compositions at 3 codon positions. GC-NSF is followed by accumulation of base substitutions to produce mature, entirely new GC-rich gene. The original gene is transformed into a new GC-NSF accompanied by the formation of the entirely new gene. By the way, how can functional protein be produced by random polymerization of amino acids? Random polymerization of amino acids in protein 0th-order structure, pre-primary structure, can produce a functional protein with a weak kinetic activity. Protein 0th-order structure, pre-primary structure, is defined as described below. Protein 0th-order structure is a specific amino acid composition in which 1. Even if amino acids were randomly joined. 2. Water-soluble globular protein. 3. With slightly more flexible structure than extant protein, can be produced at a high probability. Then I investigated on whether protein 0th-order structure can be written onto one GC-NSF or not. Let's think about the randomness more. Is amino acid sequence encoded by GC-NSF random? Amino acid sequence diversity of a small protein composed of 100 amino acids reaches 20 to the 100th equal 10 to the 130th. I have proposed GC-NSF hypothesis on formation of entirely new gene and protein, suggesting that an entirely new gene and protein has been generated from non-stop frame on antisense strand of a GC-rich gene. NSF is a codon sequence on antisense strand in the reading frame corresponding with the gene on sense strand. Can polypeptide chain encoded by GC-NSF be folded into water-soluble globular structure? 6 structure indexes of protein described below were utilized as necessary conditions to judge whether a polypeptide chain can be folded into water-soluble globular structure or not. 6 conditions for protein structure formation: hydrophobicity, hydrophilicity, alpha helix formability, beta sheet formability, turn coil formability, acidic amino acid composition, basic amino acid composition. The results show that the imaginary protein encoded by GC-NSF satisfies one of the 6 conditions, hydropathy for protein structure formation. It was confirmed that the imaginary protein encoded by GC-NSF well satisfies all the 6 conditions, which were calculated with amino acid composition. The GC-NSF out of 3 codon sequences on antisense strand is an analogous SNS in parentheses sub-N sequence similar to GC-read gene, by which amino acid sequence encoded satisfy the 6 conditions: hydrophobicity, hydrophilicity, alpha helix, beta sheet, and turn coil formabilities, acidic and basic amino acid contents for protein structure formation. The conditions were obtained with analysis of extant proteins encoded by 7 microbial genomes. Is protein encoded by GC-NSF flexible? As well known, hydrophobic and hydrophilic amino acids are encoded by codons NU, N, and NAN respectively. White bars indicate base compositions of GC-read gene, and black bars indicate base compositions of GC-NSF. Structure of an extant protein is approximately rigid when U2 content is sufficiently larger than A2 content. On the contrary, A2 content of GC-NSF is larger than U2 content. In addition, turn coil forming glycine content of imaginary protein from GC-NSF is larger than that of extant protein. This means that the imaginary protein is more flexible than that of extant protein. Can function protein be produced from one amino acid sequence encoded by GC-NSF? Degeneracy of the genetic code yields a large number of different amino acid sequences from non-stop frame on antisense strand of GC-read gene without inducing any amino acid substitution of a protein encoded by the gene. Therefore, it can be examined whether there exists a required activity onto the imaginary proteins encoded by GC-NSF of one gene or not. One amino acid sequence encoded by a non-stop frame on antisense strand of GC-read gene can generate an extraordinarily large number of different protein structures owing to the flexible protein structure. Once a weak catalytic activity for a substrate was detected on the surface of one of the protein structures, the mature protein gradually develops to a mature enzyme with a higher catalytic activity and a more rigid protein structure. Amino acid sequence encoded by antisense sequence of GC-read gene, GC-NSF satisfies 3 definitions for protein 0th order structure. Although GC-NSF encodes only one amino acid sequence, 1. It can encode an extraordinarily large number of amino acid sequences because of the degeneracy of the genetic code. 2. In addition, an extraordinarily large number of protein structures can be produced from only one amino acid sequence synthesized by expression of GC-NSF because of the flexible structure of a protein encoded by GC-NSF. Thus, it is concluded that an entirely new protein can be produced at a high probability because of protein's 0th-order structure or pre-primary structure encoded onto GC-NSF.